Chapter 14 Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by organisms to indirectly break down simple molecules like glucose, amino acids, fatty acids, etc. to derive energy to perform various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reaction. It is, therefore, evident that O2 has to be continuously provided to the cells and CO2 produced by the cells have to be released out. This process of exchange of O2 from atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hand on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following section of this chapter. 14.1 Respiratory organs. Mechanisms of breathing vary among different groups of animals depending mainly on their habitat and level of organization. Lower invertebrates like sponges, coelentrates, flatworms exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms use their moist cuticle and insects have a network of tubes to transport atmospheric air within the body. Special vascularized structures called gills are used by the most of the aquatic arthropods and mollusks, whereas vascularized bags, lungs, are used by the terrestrial forms for exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin also. 14.1.1 Human Respiratory System. We have a pair of external nostrils opening above the upper lips. It leads to the nasal chamber through the nasal passage. The nasal chamber opens into pharynx, a portion of which is common passage for food and air. The pharynx opens through the larynx into the trachea. Larynx is a cartilaginous box which helps in sound production and hence is called the sound box. During swallowing, glottis covered by the thin elastic cartilaginous flap called epiglottis to prevent the entry of food into larynx. Trachea is a straight tube extending up to the mid-thoracic cavity which divides at the level of fifth thoracic vertebra into left and right primary bronchi. Each bronchi undergo repeated division to form secondary and tertiary bronchi and bronchioles, ending up with a very thin terminal bronchiole. The trachea, primary, secondary and tertiary bronchi and the initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular, walled and vascular bag-like structures called alveoli. The branching network of bronchi, bronchioles and alveoli comprises the lungs. We have two lungs which are covered by double-layered pleura with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is close with contact with the thoracic lining, whereas the inner membrane is in contact with the lung surface. The parts starting with the external nostril up to the terminal bronchiole constitute the conducting part, whereas the alveoli and their ducts form the respiratory or the exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, cleans it from the foreign particles, humidifies and also brings the air to the body temperature. Exchange part is a site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs and the lower side by a dome-shaped diaphragm. The anatomical setup of the lung in the thorax is such that any changes in the volume of the thoracic cavity will be reflected in the lungs cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First, breathing or pulmonary ventilation between the atmospheric air is drawn in and CO2-rich alveolar air is released out. Diffusion of gases across alveolar membrane, transport of gases by blood, diffusion of O2 and CO2 between blood and tissues, utilization of O2 by the cells for catabolic reaction and resultant release of CO2. 14.2 Mechanism of breathing. Breathing involves two stages: inspiration, during which the atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air in and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure. That is, there is a negative pressure in the lungs with respect to the atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and the internal costal muscles between the ribs, help in generation of such gradients. Inspiration is initiated by the concentration of diaphragm, which increases the volume of thoracic chamber in the anterior-posterior axis. The contraction of external intercostal muscle lifts up the ribs and the sternum, causing the increase in the volume of thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in the pulmonary volume. An increase in the pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move in the lungs. That is inspiration. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduces the thoracic volume and thereby the pulmonary volume. This leads to an increase in the intrapulmonary pressure to slight above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times a minute. The volume of air involved in breathing movement can be estimated by using spirometer, which helps in clinical assessment of pulmonary functions. 14.2.1 Respiratory volumes and capacities. Tidal volume (TV). Volume of air inspired or expired during a normal respiration. It is approx 500 ml. That is, a healthy man can inspire or expire approximately 6000 to 8000 ml of air per minute. Inspiratory reserve volume (IRV). Additional volume of air a person can inspire by forceful inspiration. The average is 2500 ml to 3000 ml. Expiratory reserve volume (ERV). Additional volume of air a person can expire by forceful expiration. The average is 1000 ml to 1100 ml. Residual volume (RV). Volume of air remaining in the lungs even after a forceful expiration. This average is 1100 ml to 1200 ml. By adding up a few respiratory volume described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity (IC). Total volume of air a person can inspire after normal expiration. This includes tidal volume and inspiratory reserve volume (TV plus IRV). Expiratory capacity (EC). Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume (TV plus ERV). Functional residual capacity (FRC). Volume of air that will remain in lungs after normal expiration. This includes ERV plus RV. Vital capacity (VC). The maximum volume of air a person can breathe in after a forceful expiration. This includes ERV, TV and IRV, or the maximum volume of air a person can breathe out after forceful inspiration. Total lung capacity (TLC). Total volume of air accommodated in the lungs at the end of forceful inspiration. This includes RV, ERV, TV and IRV, or vital capacity plus residual volume. 14.3 Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissue. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in the mixture of gases is called partial pressure. and is represented by as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in atmosphere air are of two sides of diffusion as in given table 14.1 and in figure 14.3. The data given in the table clearly indicates the concentration gradient for oxygen from alveoli of blood and blood to tissues. This is table 14.1. Similarly, a gradient is present from CO2 in the opposite direction, that is from tissue to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher than compared to O2. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement membrane in between them. However, its total thickness is much less than a millimeter. Therefore, all factors in our body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from tissue to alveoli. 14.4. Transport of gases. Blood is the membrane of transport of O2 and CO2. About 97% of O2 is transported by RBC in blood. The remaining 3% of O2 is carried in dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried by bicarbonate. About 7% of CO2 is carried in dissolved state through plasma. 14.4.1 Transport of oxygen. Hemoglobin is a red-colored ion containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2. Partial pressure of CO2, hydrogen ion, concentration and temperature are the other factors which can interfere with their binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against PO2. This curve is called oxygen dissociation curve and is highly useful in studying the effect of factors like PCO2, H+ concentration, etc. on binding of PO2 with hemoglobin. In alveoli where there is a high PO2 and low PCO2, lesser H+ concentration and low temperature, the factors are all favorable for the formation of oxyhemoglobin. Whereas in tissues where there is low PO2, high PCO2, high H+ concentration and high temperature exist, the conditions are favorable for dissociation of oxygen from oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under normal physiological conditions. 14.4.2 Transport of carbon dioxide. CO2 is carried by hemoglobin as carbaminohemoglobin. This binding is related to partial pressure of CO2. PO2 is a major factor which could affect the binding. When PO2 is high and PCO2 is high and PO2 is low, as in tissues, more binding of carbon dioxide occurs, whereas when PCO2 is low and PO2 is high, as in alveoli, dissociation of CO2 from carbaminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissues is delivered to alveoli. RBC contain a very high concentration of enzyme, carbonic anhydrase, and minute quantities of the same present in plasma too. This enzyme facilitates the following reaction in the both direction. CO2 plus H2O in presence of carbonic anhydrase, H2CO3 is formed, which in presence of carbonic anhydrase further forms HCO3 minus plus H plus. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood and forms HCO3 minus and H plus. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction, leading to formation of CO2 and H2O. Thus, CO2 transported as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. 14.5 Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissue. This is done by neural system, a specialized central present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the function of the respiratory rhythm center. Neural signal from the center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in this substance can activate the center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process, by which this substance can be eliminated. Receptors associated with aortic arc and carotid artery are also recognize changes in CO2 and H+ concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm center is quite significant. 14.6 Disorders of the respiratory system. Asthma is difficulty in breathing, causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder which alveoli walls are damaged due to the respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupationally respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. The long exposure can give rise to the inflammation leading to fibrosis and thus causing serious lung damage. Workers in such industries should wear protective masks. Summary. Cells utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for transport of oxygen to the cells and for the removal of carbon dioxide from there. We have a well-developed respiratory system comprising 2 lungs and associated air passage to perform the function. The first step in respiration is breathing, by which atmospheric air is taken and the alveolar air is released out. Exchange of O2 and CO2 between deoxygenated blood and alveoli, transport of these gases throughout the body by blood, exchange of O2 and CO2 between the oxygenated blood and the tissue, and utilization of O2 by the cells, and the other steps involved. Inspiration and expiration are carried out by creating a pressure gradient between the atmospheric and the alveoli with the help of specialized muscles, intercostals and diaphragm. Volume of air involved in these activities can be estimated with the help of spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveoli and the tissue occur by diffusion. Rate of diffusion is dependent on partial pressure of gradient of O2 and CO2, the solubility as well as thickness of the diffusion surface. These factors in our body facilitate diffusion of O2 from alveoli to the deoxygenated blood as well as from oxygenated blood to the tissue. The factors are favorable for the diffusion of CO2 in the opposite direction, that is from tissue to alveoli. Oxygen is transported mainly as oxyhemoglobin. In alveoli where PO2 is higher, O2 gets bind to hemoglobin, which is easily associated at the tissues where PO2 is low and PCO2 and H+ concentrations are high. Nearly 70% of the carbon dioxide is transported as bicarbonate with the help of enzyme carbonic anhydrous. 20 to 25% of the carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In tissues where PCO2 is high, it binds to blood, whereas in alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm center is maintained by respiratory center in the medulla region of the brain. A pneumotaxic center is pons region of the brain and chemosensitive area in the medulla can alter the respiratory mechanism. Chapter